Yeah, so I will present our work not only for what we have, well, not only within CellX active, but actually we built our CellX expertise on our previous expertise, and our previous expertise, of course, is also will be also included into CellX services once it is open to Danish-wide infrastructure. Yeah, so Brick is a key node of CellX, and our intention is to bring several platforms that we already have at Brick together. I put only 3 of those here, but you will see in the very end there will be more. Together to as a kind of central coordinated service. So we used to have this core, so single cell genomics was the original core that we started, like I think 2016 or 2017, and then yeah, bioinformatics was all started even earlier. Now with CellX funding, we bring in spatial modality to our services, and you will see how we want to integrate it all. This is how currently the web page look like for our facility. It is not updated yet for CellX. Will be updated at some point closer to when we start to be open for CellX requests. And yeah, here there are 3 members of the facility. So me, but I'm like more distant supervisor of the facility. In fact, daily supervisor is Irina, so who also manages the work in single cell core. So if someone is interested to perform any project, so then you please write email to Irina, so she handles all requests. And I mean, we are open, right? We are open to any requests all over Denmark, not only from academia, also from industry. And those pipelines that are available now, and I will show you them. Yeah, you can do them. You can run them at our premises. And our idea is to have different levels of support. In fact, so it is possible to run everything ourselves. So basically, you give us the samples and then we run it. But there are other levels of support, so we can train people, and then it is do it yourself, at least for the moment. In fact, for all technologies we offer it. We will see later whether we will offer it also for some more sophisticated technologies like NeurFish. We'll see in the future, but so far all pipelines are available in do-it-yourself mode or in partial support mode, where you get few hours of support during your sample processing. So this is how CellX platform looks like. Me and Kurt, we kind of overlook what CellX is doing at our node. And as I already told, so Irina and Bente are staff members, and Linbu is computational staff, whose major efforts will be on spatial analysis. And then, of course, we have also other subsidiaries of Brick core unit, like, for example, Brick Bioinformatics, which also contribute to our work. Briefly, I will introduce our approach that we have taken to benchmark certain technologies in our facility and provide them to the users as a service. So this is kind of older graph of growing of single cell RNA sequencing technologies, and this was the first technologies that we offered in the facility. And as you also know, that it started very early with only a few cells, and then technology developed, and now we can do even hundreds of thousands of cells with some technologies. So yeah, by the end of 2021, there were more than 100 methods for single cell RNA sequencing and many combinatorial methods with other modalities. So if you can kind of subdivide single cell RNA sequencing methods in 2 different categories, one will be more focused on throughput. So that you will be able to sequence more cells, so transcriptome from more cells, but not so deep. so it would be rather shallow sequencing, or it can be high resolution sequencing, so where you try to capture all cells within rare populations, and also better RNA capture. Although technology is not yet there to be at bulk RNA-seq level, where you capture basically all transcripts from a tissue, but it's single cell, it's getting there. So you can capture 50-60% of all RNAs, of all mRNAs from a single cell, and it's basically very representative for the transcriptome. So you can get well 13,000 of genes per cell. So we focused on both of those, and I will show you technologies that we provide as a service for each of those. Of course, you know 10x, so that's what we chose for high throughput single cell RNA sequencing. And there have been several droplet-based methods, which let me just run it, which you can see in this video is based on droplets formed in a flow cell where oil is served from one part of microfluidics and from another beads and water solution together with enzymes, and you can see cells also going inside. So you will see, I think cell now, yes, going here into the droplet. And at that time, 10x was the best technology available for this kind of solution, and in fact, it's still, I think, the best technology available for this solution. At least less, you need less troubleshooting, and it's more versatile and gives most robust data. Then for the okay, let me stop it. Yes, then for the other type of technology, which is high resolution, there at least at the moment when we started to benchmark it, there were only plate-based methods for this technology. So where you would sort cells, one cell, one well, you can do it in different plate format. And here we chose a technology which is called SmartSeq2. Now there are different solutions available for this, not only plate-based, but we still stick to this one because we have quite good protocol for getting really high resolution transcriptome here. So we are talking about 12 or even 13,000 cells per 12 or 13,000 genes per one cell, which is really a lot if you compare, for example, with 10x, where you are hardly above 3,000. Right. And here's our kind of timeline. So for 10x, so first paper, which was supported by the facility, came out in 2019. For SmartSeq, first paper we supported was 2020. And for Visium, it's another technology I will show you when I will talk about spatial data. Another technology from 10x, first paper came from our facility 2022, and now we have also benchmarked another 10x kit that we provided as a service. It's called Multiome. It's RNA-seq and ATAC-seq in the same cell. It's quite expensive, but it provides very nice data where you can get gene expression and epigenetics from the same cell. And yeah, I would like to also emphasize that we got certification from 10x, at least for two technologies for RNA-seq, both 3' and 5', and for Visium, and we are going to have it also for Multiome, maybe even this year. And it's, I can just tell for other course, it's a tedious procedure to pass it, because 10x has a very thorough criteria of your data that you have to obtain, and it shouldn't be just one data set, multiple data sets. Yeah, so it was a tedious procedure, but we passed it, and yeah, we have been certified. I think around year and a half ago. Now, what we also sort of automatization. We started quite long time ago, but there were some logistics issues also with COVID. Now it's going well. So our idea was to make at least semi automatization or like keeping only little handwork for two protocols at the moment, which would be SmartSeq, which is actually very tedious protocol, takes 3 days manual handling, or 10x genomics 3' RNA-seq. And for SmartSeq, we move kind of from 3 days manual work to only 1 day. On this robot, it's a Cyclone workstation from Teton and Elmer, and with Mantis as a kind of addition to this workstation. And same for 3' RNA-seq. For 10x, yeah, we are also going to put it on the platform. I mean, from a perspective of core facility, I can really tell you that it reduces cost dramatically, because what we bill to the users, we bill time of staff that process samples, and while users, pay, similar running costs for the kits, it's fine, but the major costs, except running cost, is staff time, and it can be quite a lot. For 10x, I don't remember how many hours exactly, but I think we're at around 12 hours. We bill users for 12 hours staff work. And for SmartSeq, I don't remember, but I think it's more than 20 hours of staff work. And if you put it on robot, it's much less. Yeah, so again, it's saving. We do hope to. We are quite far into this process, and we do hope to have it done by the end of the year for both of our technologies. And of course, this will be available via CellX in the future. So this is our overview for single cell sequencing. What we have now, it's 3' and 5' RNA-seq. It's multi-ohm for both RNA and ATAC-seq in the same cell, 10x. And then this SmartSeq for high resolution RNA sequence. And what we also started to offer recently is bulk RNA-seq based on SmartSeq 2. Price-wise, Irina knows much better, but I think it's around €30 per library that is paid, and it's less than you would pay buying commercial kit. And data-wise, it looked good on the benchmarking project. So yeah, we started to offer it beginning of this year. We also already offer one spatial technology, Visium. And then in the future, we will offer more spatial technology. I will talk about them in the moment. And what we will offer in the future is automatization of SmartSeq 2 and 10x protocols, and in fact, also end-to-end services. I will talk about them at the very end. And yeah, what we also try to now see whether we can include other commercial providers. So we test, because there should be some alternative to 10x available, especially if some users are interested. Now I will switch gears to spatial transcriptomics. So this is again a similar table for technologies, or like plot for technologies, and you can see that there are different possibilities how you can write spatial transcriptomics from multiple cells, but less genes measured, or from whole transcriptome solutions, but less throughput. And again, we can subdivide spatial transcriptomics basically in two parts, like whole transcriptome, where you are able to detect all genes. However, usually you sacrifice resolutions there. It is very difficult to get cellular resolution, and even more difficult to get subcellular with this technology. Although technology is coming up, resolution here increases over last couple of years. And then there's high resolution possibility for spatial transcriptomics, where you go subcellular, so really subcellular, less than micrometer resolution. But there usually you target 100 to 1,000 transcripts, so it's more focused option. For whole transcriptome, we use Visium. It's kind of overview slide of Visium from 10x genomics, and I don't go in deep. Just that resolution is around 55 micrometers per spot, and they have multiple spots, and the size of slides that you can use is around 6.5 millimeter. This is our data, some of benchmarking activities, which was also included in this paper, and you can see here that we were actually able to annotate even individual subtypes into proper positions across here. These are layers of cerebral cortex in human tissue. Now Visium goes into upgrade into Visium HD with much higher resolution. We'll see the exact resolution that 10x will provide. Unfortunately, it is postponed again. Neither we, I think, nor even 10x salespeople know when the technology will be finally available for the user. But yeah, we expect at least maybe by the end of this year or maybe beginning of the next year. And then, yeah, resolution will be much higher, maybe not 150, maybe not 1500 as you can see here, but much higher than current Visium, which you can see here. Another technology which we will provide is high resolution spatial transcriptomics. We will use in our facility Merfish. Here's a brief overview of what it does. So basically, you have multiple routes of hybridization of your section. It can be a cell culture, it can be a tissue with different props. This so-called readout props. And here's an example of those rounds of hybridization. And for example, you can see 4 genes here, and then during round 1, you have probes for gene 1, gene 3, and gene 4. And then you will mark it afterwards as 1. And if there was no label, you mark it 0. Then round 2, you have all of them. So you mark all of them 1. Round 3, you have only a second gene, and others are 0, and so on and so on. So you label this binary code, 1s and 0s. This is how it looks as a hybridization cycle. And basically, you cleave the probes, and then you hybridize. And then again, cleave and hybridize. How it looks like on the image, it's on here or in the real image. And if so here is a single cell, and if you zoom in on one spot, yeah, here, for example, this one, it's 1 mRNA. And you can see here rounds of hybridization. You can see round 1 present, 2 absent, 3 present, 4 absent, etc., etc., etc. And so that's how you build this binary codes, and then you know when you delivered probes for each gene, right? So you know the binary codes for each mRNA, and that's why you can later decompose the image. This is how it looks like when you start to color it. And you will know where is which mRNA in which cell. This is how it looks like in a more nicer way when you start to ready to explore the tissue and have some biological hypothesis. This is also what we published together with John Moffitt, who was one of the developers of Merfish, and here we implemented our software to resolve Merfish data. And you can see, for example, so here's brain. You can see nicely how we can resolve cellular subtypes. So here they are labeled by different colors based on the gene expression by Merfish. And here is gut. So gut is very so it's a tissue with where cells are very tightly bound to each other. And in fact, it's very difficult to analyze by spatial analysis, and especially to find borders of cells. So we were able to identify cellular borders and to classify cells, as you can see here and here. So finally, with last technology which will be available at our facility and for CellX users, it's called Maxima. The instrument is already here, and Maxima goes a little further to proteins. And it can do multiplex protein measurements. I mean, they claim more than hundreds of proteins. We are not testing hundreds so far, but we are testing it between 20 and 50 at the moment. And it does look good. It's again cycles of labeling with different antibodies, and then erasure of a signal. So labeling, imaging, erasure, labeling, imaging, erasure. And erasure is done by different approaches like photo bleaching or different types of release of a dye. And Maxima has it either by photo bleaching or by enzymatic release. It has some advantages over single cell transcriptomics, as you know, and that RNA is not protein, and in fact, RNA levels and protein levels might be very different, and proteins are actually the guys who are functionally relevant in the cell, mostly contribute to cellular function. So you do want to identify protein levels. And then, of course, what Maxima can provide is subcellular protein localization and also protein assemblies and post-translational modifications, and also outside of the cell. So in single cell transcriptomics, you study what is inside the cell, but you don't study, for example, ECM, so what is secreted. It's how it looks like on a real image. And here, for example, I mean, there are multiple proteins here. I don't remember how many overall, but there are, you see, there are many here, and you can select some of them, and then you can see where they are distributed by color here. Here there are some features. Again, it's Milton claims that they can have hundreds of markers in one sample. We'll see if we can go to hundreds, but at least, you know, tens definitely, and 50. Yeah, I think it's possible. And we're getting there. It's automated. It's very easy to use. So instrument is very easy to use, and it works on fixed samples. And there are different objectives, and yeah, it has very high sensitivity. And you can also use homemade antibodies. It's more tedious. We didn't succeed with homemade so far, but at least it should be possible. So this is our overview for spatial. What we claim, what we aim to provide is first layer is exploration of your data using a whole transcriptome approaches. So we plan to use Visium HD. So we hope that it will be available soon. Then more hypothesis-driven project where you do know what to focus on, and this can be done with Merfish. And then finally, if you go from genes to proteins, so it can be Maxima with highly multiplexed, very sensitive protein analysis. And here where we are is with the time. So Visium regular is available. Visium HD, we will start to test when it will be available at 10x. And then Merfish, we are assembling it, hope start testing in fall. And Maxima we are testing since beginning of 2023, and yeah, it looks good. Finally, by the end of first funding period of CellX, we aim also to synergize the different core facilities to provide end-to-end services for CellX users. Where, so for example, as an example of full service, what we aim to provide. You can get, so you bring your cells or nuclei, and then you will get cDNA libraries, you will get sequencing done, and then at least some classic data analysis. Same for spatial. You bring sections on slides, we do hybridization, imaging, and data analysis. However, we also plan to add to our services such that you bring just a piece of tissue or cell culture that you want to analyze, and then our core facilities do everything from the very beginning. So like from tissue processing to data analysis, and same for spatial. Our core facility can section it, put it on slides, and as an output, you will get data. That's it. Thank you for listening.